Now we will explain the cardiac contractility. Okay, so about the cardiac contractility, what is the cardiac contractility? So first of all, measure the strength of cardiomyocyte to contract. How much it contract, how strong it contract, and this measure is called cardiac contractility. So this is the cardiac cells, okay, and they have a gap junction. You will explain. Here is the SA node. When SA node fired, the cardiac myocyte become activated and they contract. So cardiac myocyte, this is cardiac myocyte, okay, and they have intercalated discs, and intercalated discs there is a present a gap junction, okay. In the gap junction there is a potential etc moving toward each other. Now membrane potential, for example, first of all, how it contract. First there is a sodium channel. When sodium channel is activated, so after that sodium move in, they activate the calcium channel, and calcium move inside. When calcium move inside, okay, this calcium channel, this calcium, there is the calcium channel, this is the calcium channel, this calcium channel is they linked with, for example, this is the calcium channel, so they link with the another ryanodine receptor, okay, with the ryanodine receptor, and that ryanodine receptor is actually present in the sarcoplasmic reticulum. So if the calcium induce, okay, they activate the ryanodine receptor, and ryanodine receptor actually release the calcium from the sarcoplasmic reticulum. So if calcium is more come from the outside, okay, so it activate more ryanodine receptor, so more ryanodine receptor release, so more calcium is released from sarcoplasmic reticulum. So if calcium induce is more, calcium release is more. So you have to remember that calcium induce calcium release phenomena is occur. If more calcium induce, more calcium is released, okay. Contractility is directly depend on the concentration of the calcium. If the concentration of calcium on outside is more, so the contractility is also increased, okay. And another thing is in intracellular calcium is regulated by parasympathetic and sympathetic tract. So how can it done, okay. So first of all, intracellular calcium is by, one is by parasympathetic, another is sympathetic. So sympathetic is actually positive inotropic. Positive inotropic means that increase the calcium, increase the contractility, okay. Inotropic, inotropic, and inotropic, and one is another thing I don't remember. Okay, so first positive, it is positive inotropic, dromotropic, inotropic. These are the three things. Okay, we will explain that. So here is the positive inotropic. So positive inotropic. Now what will happen? That there is the beta 1 adrenergic receptor. So norepinephrine is released that activate the beta 1 receptor. Okay, when these activate, they actually phosphorylation occur, and in phosphorylation there is called the, this is actually the sar phosphorylation occur, and this is the channel that due to this the calcium is regulated, calcium moving out from the sarcoplasmic, this is actually the sarcoplasmic reticulum, and this is the calcium channel that regulate the calcium entry and outside. So here is a phosphoembin channel, and phosphoembin become phosphorylation of phosphoembin channel is occur. When the phosphoembin is phosphorylated, it means great uptake and great release will occur after that. Okay, sympathetic group norepinephrine is released, beta 1 adrenergic receptor is activated, and phosphorylation is occur. In phosphorylation, phosphoembin is activated, phosphoembin channel, okay, is phosphorylated, so great uptake and great release will happen, okay. So now the second thing is, this is all things are happening. Now another thing is the parasympathetic tract. What is the parasympathetic, we will explain. And you have to also remember that one is the L type calcium channel that increase the permeability, okay. So we have explained the sympathetic increase, actually phosphorylation of phosphoembin channel occur, and phosphoembin channel due to which higher uptake and higher release occur from the sarcoplasmic reticulum, and that depend on the extracellular calcium level. Now parasympathetic actually is the negative inotropic, decrease the contractility. Okay, how? When the parasympathetic activate, it activate the M2 muscarine receptor is present here. Okay, M2 muscarine receptor actually downstream all the event that we have discussed, that the less calcium channel is activated, less calcium move in and less calcium is released from sarcoplasmic reticulum and less contractility occur because there have also myosin actin bridge, and if the calcium is less, they will form less bridges will form and less contractility occur. Okay, now heart rate can change contractility, but you have to know about the drugs. There is the glycosides or foxagluaplan digitalis purpuri. Okay, and that drug is called digoxin. What digoxin do? Digoxin do the function of digoxin. You have the simple thing. I will you This is the cell. Okay, I will give the digoxin function. There are two channel. There are two channel. One is like this channel, another is this channel. So you can see that this blue channel is actually this blue channel. This blue channel is actually sodium potassium channel. Sodium potassium channel, and by this sodium move outside and potassium move inside. Okay, so when there are, so if this channel is running, okay, what will happen? What will happen? That more calcium, that sodium become high and high, sodium outside, sodium become increase, outside sodium become increase. When sodium is increased, there are another channel. What that channel do? That channel actually move the sodium inside, because there now sodium is outside is increased. If sodium is increased, it's due to diffusion, etc., due to concentration gradient, sodium is outside is more, so sodium have ability to move inside. So by this channel, this is the primary channel and this is the secondary channel. Okay, secondary active transport. In secondary active transport, sodium is want to move inside. So at but you have also to, because there if there is the charge is same, but if you want to move the sodium inside, what you do? The calcium is moved to the outside. So if this channel is activated, again this channel is also activated along this. So as a result, more calcium is moved out from the cell. So less calcium is inside, less calcium remain inside the cell. So the contractility become decreased. When the contractility is decreased by this, so if this channel is activated, sodium potassium channel, sodium potassium channel is activated, then contractility is less. Okay, if you inactivate this, if you, for example, if you block this channel, so there no sodium accumulate outside, so the sodium can't move inside and the calcium can't move outside. So this channel is also activated, so calcium can't move outside, so calcium become increase inside. Okay, if you sodium potassium channel, if you close or inactivate, what will happen? The calcium can't move outside, so calcium is increased inside. So when calcium is increased, the contractility is also increased. Okay, just like this is simple mechanism about the digoxin. Okay, you have to remember this is primary transport, and this is secondary active transport. This need ATP and this need only electrogenic, the concentration gradient. Okay, now we have discussed the digoxin. Now come here to the another thing, the sympathetic stimulation, heart rate. These all mechanism are we have discussed is okay. Now for toxicity, diffuse T wave. For example, if another thing is, if you use digoxin in high amount, there will be toxicity will be happen, and in toxicity will be the diffuse T wave will appear in the ECG. Diffuse T wave will appear in the ECG. That is the toxicity of if you use in more amount, that will show the toxicity. Okay, diffuse T wave will appear, okay, in the ECG. Now another thing is the heart rate change contractility. If heart rate is increased, action potential in the tissue is increased. Calcium that enter the cell is also increased. Sarcoplasmic reticulum of the calcium is increases. Okay, for why? For subsequent action of potential. Okay, so that's why calcium is released more and more contractility is increased. If the heart rate is increased, contractility is also increased. Another is also seen the post-extra systolic pressure phenomena. Post-extra systolic potential phenomena. Let's come here. Now, first of all, premature heartbeat. Premature heartbeat have less and normal string is actually premature heartbeat is less than the normal string because here is a ryanodine receptor have a transient refractory period. Okay, premature heartbeat. Now another thing you have to just don't confuse yourself on these things. Okay, now another thing is the sympathetic stimulation. Sympathetic stimulation is also phosphorylation of we have already explained the sympathetic stimulation. We will explain this in the other video. Okay, this will confuse you here. Okay, so sympathetic stimulation actually phosphorylation and of phosphoambin channel and sarcoplasmic calcium channel. The first thing is increase the release, uptake, storage of calcium within the sarcoplasmic reticulum. If the more calcium is released, is then storage of calcium in the sarcoplasmic is also more, and by staircase effect, like a stairs, the contractility is increased, positive inotropic. Okay. Another thing is the glycoside. We have also discussed the glycoside digoxin. Okay, that increase the contractility. Now this is all about the contractility. So thanks for watching like YouTube. Okay, and for downloading the PDF, if you want to download the PDF, see the link in the comment, etc. Thanks for watching.